Good morning, everybody here. We are starting the day 2 of the conference with one interesting talk being delivered by a senior member of this society, that is Society on Molecular Signaling, that is none other than Professor A.S. Raghavendra. We have been colleagues. He has been my senior and a highly distinguished plant biologist with several recognitions to his credit. I will only mention, other than what is mentioned here, he has been a very distinguished professor, visiting professor in Germany for almost a year, and then at University of Hyderabad, after his superannuation for about 8 years, he was on 8 to 9 years, he was on a very important positions like a reappointed professor and also as IOE chair professor, Institution of Eminence chair professor. All the outstanding contributions that he has made to the science. It is my privilege to welcome him to deliver this talk. I also would like to request my co-chair, Professor Subra, to say a few words.
Very good morning to one and all. And it's really, I would say it's not only a pleasure, but a privilege to chair Professor Raghavendra. And everything possibly has been said about him to all of you. But for the younger students who are at the back, I would like to say that in one word, if I want to think that what is the major contribution that Professor Raghavendra has contributed in the plant science domain is possibly the photobiology. And earlier, when we were students, we had few of the photobiologists in the country, Professor Singhal, if I remember, Professor Mohanty, and Professor Raghavendra. The earlier two are not with us today, but we still have Professor Raghavendra, who actually made photobiology as a discipline, I would say, in the Indian plant science. So he is a doyen in that field. To me, that is the most credibility that he has shown, and we all have learned for many years about photobiology from him, although I was a direct student for Professor Singhal and Professor Mohanty at JNU, and those days we used to see Professor Raghavendra coming and at JNU quite often. And therefore, I know him from my student days, and he's also, on top of that, my last point would be, he is very, very kind to all the young colleagues, to the students, and he has made a niche, and we all respect him like anything. So with that, the stage is with Professor Raghavendra now.
That's for changing. Thank you so much, Professor Appara and Dr. Shubra. Very nice friends for a long time. And it's also my great pleasure to acknowledge the fantastic support from this university in several ways. And specifically, I have to thank, because as a president of the society who was involved in planning, it's a double pleasure to thank once again. As you can see, can we go? Yeah. You will be quite intrigued by the title. You may ask some of you, I think there are students from lower level to experienced plant scientists and experienced animal biologists. How come plant can be indicated with having neurobiology? So the first part of my talk, I wish to point out that yes, they don't have neurobiology in the strictest sense. When you say neurobiology, you think there is a brain and there is nervous system which will go around the body, and then, so for example, when you are walking and if you feel that there is sun, you just run into the shade. That is, the brain decides. So the skin detects the heat, sends the message to the brain, it's time you protect yourself. So the brain, what you call as, sends a message to legs that you should move. Okay, similarly thirst. When you have thirst, you have a desire to drink water, and this is because there is a central nervous system which coordinates to receive the signal and respond. If you come to plants, yes, they don't have a nervous system, but they have something very similar. They have a system where plants can detect signals, then respond properly, and then it becomes a challenge because they cannot run away from problems. If there is a problem with temperature, water, pathogen attack, they have to stand there and face it. And so this is one lesson we have to learn from plants, how they become resilient with their own limitations. And so my first talk, I'll try to tell how they sense and how they respond quite interestingly. And so now if you see, no, it doesn't work. Yeah, this is the something like summary. Human neurobiology, as I have already indicated, yes, I think Professor Dr. Balram also was telling that we have 5 senses. We can see, we can smell, we can touch, we can hear, and we can talk. In fact, I sometimes give a talk indicating how plants have all these senses. So today, quite likely I will touch upon a little bit of, how they touch, how they sense the attack of, say, like pathogens or insects and so on. Yeah? Yeah, sorry, I think one of the slides ran into problem. So this is what I mean. I don't know how many of you are familiar with this plant. This is called Venus flytrap. It's given the name flytrap with a purpose. I think as we go, I will also show you how it does it. It can trap small insects. This is a small plant. I think it doesn't look as big as this, although it is amplified. And so when a insect falls into this lobe, we have 2 lobes here. Look carefully, there are 3 to 4 hairs. They are the, what you call as touch sensitive tissues. So if the insect touches at least 2 of them, the leaf responds. And this is something which it cannot waste energy, because closing takes very quick time. In a few seconds it will close, but opening will take almost like 24 to 48 hours. And this, they do it not very, very repeatedly. This plant actually grows in a place where there is not much nitrogen. So sometimes they depend on these insects which are useful as a nitrogen source. But as I said, oops. Yeah, this is just to say how plants sense light. I think you all know. There are beautiful examples how the plants sense light, and they sense also the sun movement, because here you can see sunflower and geranium, and then A plant growing towards light is a very common sight. You must have all seen it. And as you can see, how it is done is, for example, when you have a tissue and when there is light, I think you all know that plants have these auxins which are responsible for cell elongation. So the side where the side where it is exposed to light, the indole acid gets what you call as degraded, and that's the reason the plant exposed to light becomes not so elongated, the other side gets elongated, so it bends. And if you look into the pigments, what they are involved in this, I think you all know that there are different kinds of pigments. Although plants do not have any eye, they can detect all kinds of pigments. So what you see here is pigments which are phototropins, cryptochromes, phytochromes, and these are the ones which are involved in development. And photosynthesis, you all know, plants are able to make photosynthesis because there is a lot of chlorophyll protein complexes in chloroplast which can sense light and convert solar energy into electrons, excited electrons. They go and make the photosynthetic electron transport system, and so they are very, very capable of absorbing a lot of light. They absorb light sometimes too much, so they also have protective mechanisms to protect themselves from excess light. Yeah, the next point is they also have the ability to smell. For example, yesterday we were talking of jasmonate signaling and so on. And for example, this is something which is quite interesting. Yeah, you can see jasmonate is a methyl jasmonate particularly is a compound which is very, very volatile and smells nicely. You know, the name has come from jasmine, which is a very typical flavor. You all know how nice it smells. And when plants are affected, when they are exposed to something like, either fungi or bacteria, as you can see, fungi or bacteria, they have to enter the leaf tissue through stomata. So these stomata, they respond immediately when something like this comes, they close it. And one of the reasons is the presence of methyl jasmonate close by, and then that's the reason. So they use the attack to trigger jasmonate content, and this methyl jasmonate in turn closes stomata. So we have a leaf responding to the attack by the pathogen. Pathogen is not allowed to enter, but then the beautiful part is pathogen can also fight back. It gives a compound which is quite similar to another compound which makes them open. So this is a biological warfare which goes on between plants and the microbes. Yeah, this is what, close up. You can see how the pigments are entering. These hyphae are growing through this stoma, and here we have a lot of bacteria trying to enter, and the plant has to do it. So in addition to methyl jasmonate, there are other elicitor compounds. A lot of them are volatile, which are produced when you have an insect attack. And so what they do is they produce compounds like methyl salicylate and a few other compounds. And these compounds, they repel insects, so they just run away. And then after knowing this, people are now trying to develop, like methyl salicylate sprays to drive away plants. I think methyl salicylate, you may be knowing, in many of the what you call as pain ointments, if you have a pain in the muscle, you normally apply this methyl salicylate containing creams. So it's a very common compound, easily synthesized. So the point I am trying to make is plants can sense the fragrant volatiles induced by pathogens, like for example methyl jasmonate or methyl salicylate and even glutamate. And what they do is when the plant detects these compounds, they normally respond. And I think I should tell about calcium. I think there was some problem with that particular slide. All of us, humans, we know that if you want to raise your hand, you will raise your hand. So where from the signal is coming, it's from brain. And when brain decides that hand has to raise, it will use the muscles. If you have, see if these muscles are straight, your hand is straight. And if they decide the muscle should say like lose water and shrink, it will come down. And this kind of thing happens also in plants. And so this movement of muscle, how it stretches, is sensed by calcium signals. And once you have calcium, it changes the content of these osmotically active compounds like sodium, chloride, and so on. So in animal tissues, in response to calcium, the muscles are allowed to either contract or expand. And in fact, it happens also in plants. The plants, they sense fragrant molecules and respond by putting out calcium, and this calcium makes the leaf to fold or to show the See here, as you can see, an insect is biting, and the moment it bites, the plant responds. So these leaves, they are loaded with calcium sensitive dyes, and then what you see here is a beautiful movement of calcium through the vascular system. And it's also a method, the plant is what you call as warned that there is danger in the vicinity. Similarly, this can be done by using external simple compound like glutamate. Now you see there is glutamate applied here, immediately plant responds with calcium waves. So when you have this calcium release, it modulates a lot of ion flux. It allows a lot of potassium to go in. I think you're all familiar how guard cells, they respond when you have potassium. So they push potassium when it is needed, they take out potassium when it is not needed. So very normally when you have more calcium, the protons are sent out. So protons are, potassium efflux happens. So these channels are modulated in such a way that when it is required, they actually send out. Yeah, I think this is not very important. There are also other volatile compounds, they do this job. And this kind of thing becomes almost like systemic, and that's what makes plants very interesting. Although they cannot move, they can do it very nicely, as you can see here, because there is an attack by pathogen here. Initially there was some response, and this one now will spread whole in the canopy. So it's not just that leaf or a particular branch, but the whole thing can be transmitted continuously through, see, there are a lot of leaves around, and most of this calcium increase happens in the vascular system. Yeah, this is what I am saying. Plants can feel touch, and I will show you now an example of Dionaea heat trap and then also Mimosa pusica. So what they do is, I think I should, yeah. See what you see here is a Mimosa pudica. If you touch it, it starts closing leaf by leaf, leaflet by leaflet. And the reason why it does is, there is, the moment you touch on the leaf, you can see here, the cell senses the touch. So there are touch sensitive calcium channels. They start acting and they release a lot of calcium inside. And when they release calcium, what happens is, this part becomes, they lose ions, so they fold down here, as you can see. So the moment you have calcium, there is a loss of potassium here in the cells, becoming this pulvinous, and the leaflet goes closed. And that's what you can see here. So we are monitoring calcium, again because of touch. So there is a touch here at the tip. You can see that white fluorescence is indicating calcium. So this calcium increase is sent through the plant system as waves. So quite similar to our nervous system. And this can be shown even with insect bite, same principle. So as the insect bites, there is a rush of calcium. So this kind of movement serves two purposes. One, the insect is scared. The second one is it tries to protect all the leaves as much as possible. Yeah, I think we will come to the stomata. The stomata also, they are very important, as you can see here. Oh, sorry, I think something happened. Yeah, this is a summary slide. As I said, plants can see, plants can smell, plants can feel the touch, plants can hear, and plants can talk. I think the other topics I will be elaborating. Oops, I think I messed up. ఇది ఏదో అయింది యాక్చువల్లీ ICMS అనే ఉంది happened in between, I don't know. No, this slide has something happened. 25.2, I think I am looking at the wrong one. Can I have my, I think, the bag on journey. Not how I will try to use that. Somehow it's got messed up. Avi desktop, ఇది డ్రైవ్ ఏదో ప్రాబ్లంలో పడుతుంది. No, this is gone. You got it. Something happened. No, that's fine. Now I got it. Some mix-up. Yeah, I'm sorry. I think I now got the correct set. Now, as I said, plants can sense and transmit the signal, And Dionia muscipula, which is also called as Venus flytrap, is a beautiful example, as you can see. And as I already said, this is called trap because it traps small insects. So this is how it looks. See, for example, here we have two lobes which are normally open, and when they get into the touch by No. I think there's some problem in the computer. Yeah, let me try whether I can show this movie. I think some virus has come when I came here. Okay, this should be all right. Let me, what you call as, get your attention. See, this is what I call that, it's a close-up of, again, Venus flytrap. Now you can see there are normal leaves. These normal leaves can make photosynthesis and supply as much food as possible, but sometimes they will be short of nitrogen, and when they are short of nitrogen and when they get in touch with some of these insects, they start eating insects. That's why it's called insectivorous plant. And now if you see carefully what is happening, see here you have say something like 3 lobes, and there is something like a set of 3 friends coming in, and now you can see it's already, one of them is already trapped. Now if you go a bit later, so there are 2 flies which are still around, and they will start looking for their friends. So one friend is already trapped, so this fly is trying to locate the friend, and also the other one also is close somewhere nearby, and this is still trying, but meanwhile the other friend comes and gets trapped. Okay, but our original friend doesn't lose patience. It will go on trying, but unfortunately as the time goes, plant becomes more alert, and unfortunately it also gets trapped. Gone. Yeah, this is a beautiful system. As I said, closing will be quite quick. It happens with a few seconds or a minute. Then, but to open it, it will take a lot of time, and plant cannot afford to do that. And as I said, you look carefully. There are 3 trigger heads. In the other slide, I have shown you 4 trigger heads. And what is happening is, when you touch this hair, when you touch this hair, there are touch-sensitive cells, and they emit voltage. I think you all know that we are all negative. Do you know that we are all negative? Anybody who doesn't say no? We all have an average negative potential of about minus 70 millivolts. So this is normal in a living cell. And unless you have this negative potential, negative electrical potential, you don't have life. And if somebody, you all know this cardiogram, they put electrodes and they try to detect the signal from the heart, because heart has a beat and it emits signals. The heart emits signals, and that's how it beats. And if you try to, what you call as, compare, the heartbeat is actually not very quick, but it's kind of slow. It's about 200 milliseconds you get each peak. Whereas muscle is very quick, I think you know, if you hit some stone, you get injury, your body responds very quickly. So this is much faster than the heartbeat. Then the muscle contraction takes a bit longer. So that's what you see here. This is about 1 millisecond, this is about 3 milliseconds, heart is about 200 milliseconds. But if you come to the dyspnea or venous flytrap, it's much slower. As you can see here, it takes a long time to respond, but then to recover from it again, it also takes a long time. And that's what is indicated here. And it's necessary to touch both the signals, otherwise it doesn't respond. And then after closing, this is again very interesting how plant adapts. Now you see here, after closing, the leaf has a different function. It becomes like our stomach. So the moment it closes, it starts producing a lot of digestive enzymes. That's what is all listed here. And the digestion is so perfect that when the leaves, they open after a few hours or after overnight, the trap which has this fly or small insect inside is all completely digested. Very efficient digestive system, is much more efficient than our own. So the cocktail of enzymes digests the prey very efficiently and nothing is left. And a very similar touch sensitive movement happens with this beautiful plant. I think there will be a lot of them around, Mimosa pudica. It says touch me not plant because it, well, not that it doesn't like. If you touch, it thinks that there is somebody harm coming and they close it. And as I said, this, you have already seen a small movie, how this calcium signal is transmitted from the point of touch to the base of the petiole. And the reason is that this base of the petiole has two types of cells. We have cells which are rich in potassium, and then the petiole becomes erect. And if these cells, they lose potassium, it becomes droopy and falls down, and that's what happens here. So these cells, the movement of potassium and calcium will make whether the plant or this part turgid or flaccid. And this is decided by the calcium. Calcium is the master signal which does this job. So this drooping movement happens at the leaflet, but later it happens at the petiole. I think this is what you have already seen. I think in my previous slide, it got, there is no need. Yeah, this is what I was telling. If you have a system where you can measure calcium, the moment you touch here, it starts producing calcium waves. And the moment it reaches each base of leaflet, the potassium ions, they are sent out and the leaf closes. Yeah, now I think I will show you something which is definitely a lot more interesting. I have taken this from YouTube. There is a place called, it is a TED channel which makes a lot of these things. Can I have a mic? Don't worry, if this can sense, that's fine. Don't worry. Okay, I think I will explain what's happening. So now we have Greg who is explaining this phenomenon, and so he will start telling about how plants are different. And incidentally, TED is a very interesting site. You get a lot of interesting pictures. So he says he's a neuroscientist, and he tries to impress upon us that plants have a nervous system and how they respond. So it all started with a lot of, these people, they have these signals being detected from their bodies. In fact, over a period of time, you can see he has the place where you can put electrodes. And this slide says, if there is a question to ask if animals have brain, you all agree, yes, they have brain. But if this question is asked in relation to plants, people are usually hesitant. Some people may say there is no brain at all. It's not like that. There is no brain, but there is a similar nervous system. So that's what this video tries to show. And now he will try to demonstrate by looking at the electrical signals. So he has a system where he first measures his own electrical signals from his own body. As I said, all of us have negative signals, and they sense touch, and they can detect this. And of course, as usual, our central nervous system is the one which coordinates as a, like a processor. And this movement of plants in relation to light is well known. And then what happens is, I think I will try to push it a little bit. Yeah, now you see he is now connecting his skin with electrodes, and he will try to show on the screen how it responds. So the moment you connect, you can see the signal. There is a kind of small signal, and you can see these undulations because the heartbeat is not a simple signal, it goes on repeating. And of course, doctors look at EKG and say that you are fine, you are not fine, depending on the pattern of these signals. Now, if you try to use this kind of detection and look at, say, like Venus flytrap, see here we have what you call as Mimosa pudica, which is very big. And the Venus flytrap, they are very small, as you can see here. So he will use one of these. This is Mimosa pudica, and if you touch, it will respond by what you call as folding itself. First it folds, and then it folds at the base. So here it folds at the base. So you touch it a bit stronger, yeah, it just folds. I think it's very common, people, particularly children, they play with this plant all around, and it's quite interesting in terms of science. So although it's named as touch me not plant, but it has a lot of science in it. Now if you try to go and then try to measure the signal, why it's happening. So he has already set up a system where the leaves are, connected, as you can see. The leaves are connected with a very sensitive device to measure voltage, and of course you need, some kind of earthing. So earthing, he has already connected it. So now you see what happens when he touches. Actually, you have a quite enlarged, very, very high signal. But then they are not very quick, they are a bit slow. And so if this is done in case of Mimosa pudica, that means Mimosa pudica is able to detect touch and convert into electrical signal, then convert into what you call as calcium waves. And then these calcium waves are making potassium to move out, cell becomes what you call as flaccid, and it cannot hold the leaf anymore. And now a very similar kind of thing happens with Mimosa pudica. So then with this fly trap is very small. And as you can see here, see, these are all fly traps. It's very small, and he will try to go and then connect and see what happens in terms of voltage. So these are the triggers. You have 2 of them, 3 of them here in each lobe. You touch one, it doesn't close. That's what I said. You have a signal, it responds by making the electrical becoming more negative, but it doesn't stay and it doesn't close. You need at least two of them coming in succession quickly. So unless you do that, the leaf will not close. And this is a kind of protection. You may have a big particle falling onto the leaf does not respond to them. So this is what happens. Now, if you try to touch two of them, then the signal you can see very good. I think he will do that. Yeah. Okay, now he will try to touch more than one signal in quick succession, then the leaf responds. Nothing happens. Very clever. Now let us try and touch more than one. Yeah. See, that's what happens. When you touch more than one, the successive signals will make the leaf to close. Now the funniest part comes. Now is this possible that the signal is transmitted through the leaf? The answer is yes. Now he will try to connect Dionia field trap. See here we have a system where you have this field, fly trap and connected to Mimosa pudica, and if you now touch Dionia with the same system and try to generate the signal, and then what happens? Yeah. Now he is trying to touch fly trap and make it close, and Dionia and Mimosa, they are connected. So the signal from Dionia actually goes from here, is the electrical signal is transmitted into Mimosa, which means that the signal can be transmitted. So this is a very nice indication that the signal is transmitted through the plant, signal is transmitted across the plant. So it is the electrical signal that is detected in principle. Well, if you want to buy, you can buy. It's available in Amazon, but very difficult to maintain. Before I go into the second part, I should also mention something about Sir Jagadish Chandra Bose. You all know he's very well known for his contributions to plant physiology in general. During his time, you can see he's almost like 400 years old. Yeah, and he has been studying plant feelings for a long time. Now, if you see one of these slides, see here, you see, this is what he has done to record how plants is in. So, with very sensitive kind of system, he has measured a lot of signals from the plant. He said they are like us and they can sense the signals, they can respond, they can transmit, and so he is now recognized as the father of plant neurobiology. So this topic of plant neurobiology has now become very important. People are studying and acknowledging the effect, the contributions of Jagadish Chandra Bose. Is it okay? Shall I stop or you have some time? Okay, let me see if I have something interesting. Yeah, I think let me just comment. When you talk of stomata are small, minute pores on the leaf surface, and very nice. When you look at them under a scanning electron microscope, they look very good. And this is a pair of guard cells which make stomata, and these are very, very dynamic. In response to a lot of signals, they can take in potassium, they can send out potassium, and this is master regulated by calcium. So first they respond with calcium and then they push potassium out. And this is done by, yeah, this is what I said. In general, the signal is sensed on the surface by a receptor, and this receptor sends the signal in such a way that the calcium is increased in the cytosol, and increase in calcium starts doing a lot of other things. So we have been studying this for a long time and quite in detail. I think I will just try to show you 3 movies where we can do this. Well, sorry. Yeah, for example, you can see there is an open stomata. The guard cells are very turgid, full of potassium and chloride, and this can happen when there is not much of calcium around. Calcium levels are low, potassium levels are high, chloride levels are high. When there is a problem, if they want to close, the calcium is allowed to enter by electrical signals. The moment you have high calcium inside the guard cell, it starts sending potassium ions and chloride ions out. So the regulation of these ion channels by a master regulator like calcium will make whether guard cells are turgid or flaccid, and it does it. Now this is what happens with calcium. The moment you have a signal like abscisic acid, the stomata, they close, the calcium starts increasing, and this is the one which makes a lot of other changes. And you may ask whether this can be checked in the lab. Yes, we have been trying to see this. And for example, we use a lot of Arabidopsis and peas for this kind of experiment. You take the strip, strip is single cell, it's very easy. You can look at it under microscope. You can push fluorescent dyes inside. So you can use a dye which is sensitive for pH. You can use a dye sensitive for ROS, and you can use a dye for sensitive for nitric oxide. And so this, for example, epidermal cell, which is loaded with DCF-DA, an indicator of ROS. As you can see chloroplasts which are red, and ROS indicated by this. Yeah, this is what I said. You can take this strip, put it onto the slide, attach it to the slide, and make the incubation medium in such a way that the stomata, they either open or close, and use a good microscope and start making pictures, and these pictures can be stitched into a movie, what you call as, yeah. See, this is a movie which actually took about 90 minutes, but the actual time here is about 40 seconds. So they are open now, and we apply abscisic acid at the beginning of treatment. After some time, they start closing. As you can see here, for some reason, only one side, it's losing potassium and closing. As down also, there is something here. And then the other one responds. So what is happening is, within a few minutes, the stomata, they lose potassium, and then they close. So here, for example, the summary is that when these epidermal strips are exposed to ABA, abscisic acid, the stomata, they close, and this closure is happening around 40 minutes. In comparison, if you look at pH is much faster, and as you will see in this movie, which is done and recorded in the lab, and you can see the pH sensitive fluorescence increases quite quickly, much faster than the stomatal closure, but pH doesn't really increase and stay there like that. It again comes down, and so this is what is represented here. Cytosolic pH increases up to 12 minutes transiently, then comes down, and then we have also a measurement of ROS, and when you look at ROS during the same time, you can see, if you see carefully, the increase in ROS takes some time, not as fast as pH. So after some time, the levels of reactive oxygen species, they increase and stay there. Actually, after some time, not like pH comes down, whereas here, the increased ROS stays for a very long time. Yeah. So these measurements can be quantified, and that's what is shown here. If you start measuring and then trying to put in relation to untreated one, you can see, and this is what happens. pH starts early, goes down. ROS starts late, but then stays there. So that's what is happening. Stomata need about 40 minutes to close. pH needs about 12 to 15 minutes. ROS needs about 30 minutes to close. So that's the kind of thing. And this is summarized here. Yeah. So what is happening is when you have a signal like methyl jasmonate, abscisic acid or elicitor, there is an increase in pH, and this increase in pH is causing an increase in cytosolic pH and then increase in ROS, and the increase in ROS again makes calcium to increase. When calcium increases, potassium moves out. Potassium, chloride, malate, everything moves out. The guard cells, they lose their turgidity, and then this stomata, they close. So this kind of events, as you can see, actually are very common, whether you have a hormone or elicitor or I will say injury, it all happens. So effects of several triggers are quite similar. This sequence of calcium, pH, ROS and calcium remains the same. In some cases, we also have nitric oxide, but nitric oxide role is not very, very clear. So I think I will leave this slide just for a while because as I said, we have a lot of signals which make stomata to close and mediated by several factors. All of them, they end with increase in calcium, increase in ROS, increase in nitric oxide. And this calcium master regulator, what it does is it modulates a lot of channels. It will not allow potassium to come in, it will only make potassium to go out. And similarly, chloride to go out. And then that's the reason the stomata, they close. So, thank you. Well, I'm sorry, I think there was some mix-up of slides.
Thank you very much, sir. We know there was some disturbance in the initial part. That's where the lecture got prolonged a little bit. But still, I would say, really it is very interesting to see that a concept which has been there but nobody talked about it before in this terminology, that is plant neurobiology, explaining how the different senses of the plants can work in absence of even a brain and a CNS kind of a system. I'm sure it must have triggered some questions and some queries in the minds of the audience. We can have a couple of questions, although it is a little late. Open for discussion.
Sir, very interesting, Professor, your talk. Let I ask one question. Is there any control system in the plant?
Pardon?
Control system, like brain?
No. That's what I said. Plants do not have brain. Plants do not have an organized nervous system as we talked in case of animal tissues. What is happening is electrical signal is, yes, generated, and people believe that this electrical signal is transmitted through cell walls. So you don't need even a living part. Cell wall is a very good conduit for electrical signals. And then I have to also tell, for example, if you take trees, they have non-availability of water in the roots. If water is not available, and if it takes 2 to 3 hours to say leaves they should close and try to conserve water, it has no meaning. So, but then the root system can detect drought and send the signal within a couple of minutes to even canopies which are as high as 100 meters. And this is by hydraulic conductivity of signals. Yeah. But there is a system which can transmit the signal, not necessarily nerves.
Sir, just one question. We have learned about the starch-sugar hypothesis also. And now how can we connect this potassium regulation as well as...
Yes, actually good question. I have been telling the importance of potassium and chloride and so on. If you take leaf in nature and look at stomatal opening, normally stomata they open in the morning. Morning they don't need starch because guard cells they don't make starch. So in the morning until about say like 1 p.m. or 2 p.m. they will use potassium and chloride as the major drivers of the turgidity in the cell. A lot of these ions, they go into vacuole. The moment they go into vacuole, your osmotic potential becomes more and more minus. Minus means like a sponge. It tracks water as much as possible. But at the end of the noon or late in the afternoon, they make enough starch and sugar. So a lot of this sugar starts accounting for osmotic strength in the guard cell. Not necessarily they make, so a lot of mesophyll sucrose also can come. See, guard cells alone will not be able to produce as much sucrose as is needed for this osmotic potential. Mesophyll contributes at that time. So later in the day, they become important.
Sir, I have one question. Like in case of animal digestive system, do the plants have any protective system against the enzymes? Because they also undergo the process of digestion. How they can get the protection against such enzymes during the process of digestion?
No, I could not understand you. Can you repeat the question?
You just mentioned about the insects are trapped and they are subjected to digestion so that no traces are left inside. No traces are left inside. How the plant itself can get protection? Because our digestive system has got, for example, the mucosal lining.
Well, of course, animals are known to move away from danger, but plants do not have that privilege. They have to stay there and protect themselves. Sometimes they are successful, sometimes they are not. And there we have the importance for a lot of these secondary metabolites. I think yesterday we were talking. Unlike normal tissue like humans, they make all kinds of secondary compounds which are all involved in protection at one time or other. So they have a lot of backup. You know, when I talk to my students, when I am giving talk, I always say, plants are example where you have an alternative for everything. You take photosynthesis, you have not only C3, but you also have C4 and CAM. You take respiration, I think we all know, respiratory electron transport is inhibited by calcium. So you know that cyanide is a very important poison, and if you have cyanide, the cytochrome pathway is stopped. But dry on plants, happily they respire because they have again alternative oxidase. Then you go to glycolysis, they have a normal glycolysis and they have also the oxidative pentose phosphate. So what I am trying to tell, plants by evolution, they have developed a lot of alternative systems. So if something doesn't help, the other thing takes up. So they try multiple ways of protecting themselves. So that's what makes plant. Yeah, please.
I have a discussion. Very fascinated about the concept, and I was impressed about this mimosa pudica from my childhood. The question is this: the signal transduction that's happening in the mimosa pudica leaf, if you touch, suppose you touch in the middle of the leaf, will the signal move both ways? The signal transduction is unidirectional or multidirectional?
Yeah. The tip is very sensitive. The explanation is that they have higher density of calcium sensitive channels. And then if you want to have this same magnitude, you have to make a stronger touch. And that's why with a small touch, the leaflets they close. I think somebody can play in the nature also.
I have done that experiment. I have touched the middle of the leaf and that closes.
It will,
But nothing happens.
If you touch the middle of the leaf,
yeah.
Normally transmission is
Those two will
towards
close.
the base,
Okay.
but it will go this side also.
If it's a little strong, it will go both ways.
Oh yes. Oops.
Okay, I'm sure all of us enjoyed this wonderful talk as the first lecture to start our day 2. And I would like to request every one of you to put your hands together to big applause for the wonderful talk. And also I request my co-chair, Subra, to close this.
So I think this speaks about, not only a researcher, but a teacher. So the way nicely he explained to us about the neuroscience, which no one as a plant or animal biologist or human scientist have ever thought, although Jagdish Bose has told this many, many years before, but today we know it is a reality. And it's a very vibrant area that is coming up worldwide. We still do not have that kind of labs in our country. So maybe Professor Raghavendra's opening talk today has encouraged all those who are sitting behind and who would be actually the leading light in next 5 to 10 years, who would take the Indian science forward. And I think all of you must be very, very excited to know about how we can connect and how plant can sense all the beautiful senses that we can, as a human, we can also sense. And the whole science to decode the plant sensory organs, and the rest is on. Really, we saw again a teacher in you. That's what I was talking to Professor Apparao, and thank you all.
Thank you, sir, for an insightful talk. May I now request Professor Apparao Podil and Dr. Subra Chakraborty to present the memento to Professor Agepati S. Raghavendra.